Today I have been advised to share my experience of the latest bioabsorbable stent, which came after the BVS experience initially with the Abbott stents, but now this is the Mary's 100 stent, which is a new one, which is now available at the moment to be used in India. So this is the only stent which is biodegradable, scaffold, which is available in India. So why are we talking about bioabsorbable scaffold? Because this is what the problem is, that the arteries get caged and we want to uncage these vessels because we want these vessels to grow, we want these vessels to become to normal size. So all this is important in routine PCIs, in left main, everywhere, multi-vessel bifurcation stemming. So in all these indications, this technology can be of help. So what happened? We were using the BVS, which was available from Abbott, and then the problem happened in many places, and then the studies did not show so good data. All this probably these are the probabilities that the large strut thickness, the bulky, high-profile device, compromised radio opacity, and the storage conditions, increased dependence on imaging, overlapping not being ideal. So all those problems kept on accumulating, and that led to the I must say the almost the extinction or the demise of the first generation of device. But now we have the second generation device, which is the Mary's 100, which is a hybrid geometry compared to the multi-link design of Absorb. And you can see that we are comparing with the metallic stent also. And here you can see that the stent scaffold design, it is made of 100 micron thickness compared to 150 micron thickness of Absorb. And here the crossing profile is only 1.2, same as a resolute integrity. It has 3 markers at the edge, and these 3 markers show up very well on the fluoroscopic image. Whereas in the Absorb, it was 2 markers at the each end. Here we have 3 markers which are placed circumferentially, so we can have a complete profile. Now, the size matrix is that they are available in 2.75 to 4 millimeters, starting from 16 to 40 millimeter length, whereas the Absorb was not available in such big sizes. And the storage condition, this is the very important thing. It can be stored at room temperature, not to be stored in the refrigerator. Now here, this is the complete picture of the stent. You can see 3 markers, and then of course the geometry of the whole stent. The scaffold backbone is made of PLLA, 100 micron strut thickness, and the side branch access is also very good in this device. The drug coat of sirolimus is 1.25 micrograms per square millimeter. So this is how it is. And the balloon overhang is very short. So abrupt balloon shoulders help in having a low dissection chance of at the edges. And this is the first study which we did, the Mary's 1 with 108 subjects. Then the Mary's Extend was done with 64 subjects by Dr. Abizade. And then now a study is going on by Dr. Colombo, which is an ongoing study right now. And then, of course, the new study which is being planned is the Marathon randomized control trial, 2:1 randomization with 1,875 patients. So this is the entire situation of the studies going on. And here is the completed data of 108 patients with 3-year follow-up. And you can see the results are really very, very good. And this is a very, positive data in favor of the Mary's stents. So let me now quickly show you some cases where we have done this. So this is a kind of a simple case, but certainly this is a patient where, we used the Mary's stent, and you can see that first we do a pre-dilatation. And once there is a pre-dilatation, a good pre-dilatation, almost going to the similar size of the vessel, you can see, and then deploying a size, because now the stent comes with 2 balloons also. So there is no problem in dilating and post-dilating at the same size. And the post-dilatation is 0.25 higher than the size of the stent. So this is the post-dilatation, and you can see the final result is very nice. You can see the 3 markers at the top and 3 markers at the end. And similarly, in these kind of even smaller size vessels, using a smaller stent here after the pre-dilatation and the post-dilatation, and you go to high pressures and see a good result. So And this is at the branch. So you can see that the branch almost at the ostium of the OM, the stent is positioned and going into the mid part. And looking at this little tortuous artery, again it goes with ease, pre-dilate these vessels, and then we deploy the stents and post-dilate to high pressures. So basically, it moves quite easily without any problem, even in tortuous segments, even in distal segments, and we get good results. And here is another patient with the in-stent restenosis, which we did. And you can see that the in-stent restenosis of a metallic stent can now be, there's a new indication of using a Marie stent, where we pre-dilate well, do a good expansion of the, you can see a huge expansion of the stent. And once the stent is expanded, we do a OCT, we are doing a OCT. And after that, we put a Marie stent here. We are putting a Marie stent with the stent boost. And you can see that 6 markers, 3 up and 3 down, and the stent is very well dilated, go to high pressure post dilatation, an extremely nice result. So this is the way it looks. You can see the result is so nice. And this is the OCT picture of these two. This is the pre-OCT and this is the post-OCT. You can see, you'll start seeing the stent struts of the first metallic stent. And now you will look at these small cages, which are absolutely stuck to the vessel wall, very thin, very narrow. And this is how it looks on the OCT. These small, tiny boxes, rectangular boxes. And these are quite well stuck, no malposition, nothing. And we leave the patient on medical therapy now, and he is doing very well. So these are the kind of outcomes we are getting. So the Maries 1 trial, the first human evaluation of novel second generation 100 micron stents, high acute success, good long-term clinical success up to 3 years follow-up with a low MACE of 1.87% and no stent thrombosis. So this is something phenomenal. We are very positive about this whole thing. And the Maries Extend also had the same MACE, 1.6%, done in a different country, different operator, and 0 scaffold thrombosis. So 2 data, very positive. And serial QC analysis show a very low late lumen loss of 0.186 months, suggesting high efficacy on inhibiting the hyperplasia at late follow-up. OCT analysis demonstrated sustained mean flow and virtually complete stent coverage at 6-month follow-up. So these cases, we have seen, and they're really looking very, very positive and leads to a great future of BRS technologies for percutaneous coronary.
Okay, thank you, Praveen, for your very illustrious discussion. One question regarding the maneuverability of the stent in tortuous lesion and how it acts, and it's rather how beneficial it is in the moderate to severe calcified lesion.
Okay, so very, very interesting questions, Dr. Banerjee. One is that in tortuous lesions, as I showed you, it moves in quite easily, better than the absorb, and the profile is 1.2, only same as integrity. So not much problem, but definitely we'll use all those methods of crossing the stent as we do for a metallic stent. Sometimes we need a guide liner, sometimes we need a buddy wire. All those things can be done, and this moves nicely. The second is about the calcific lesions. Definitely, now that is why the stent is coming with 2 packed balloons. There is a package, and where we can go to high pressure NC balloon dilatation for calcified lesions. We can do a cutting balloon. We can do a shock wave also sometimes, and then we can implant these stents in these lesions. It is better to debulk the lesion, prepare the vascular bed. That is the most important thing. As you said, this is very, very critical.
Thanks, Praveen. Any other questions or comment on the issue? Any panelist or discussion? If there is no question, I don't find in the chat box there are 2 questions anyway. Praveen, I am just.
Thank you.
Okay. Thanks for your presentation of the study. Now.